Dr. Zakeel from CSIRO will share her expertise about the epidemiology with the topic plant disease epidemiology, case studies on tracking.
Oh, di layar ibu. Di minimize aja Bu.
Okay. So today, Zakeel or Dr. Zakeel will talk about the plant disease epidemiology, case studies on tracking and managing pathogen spread across landscape. So before we start, I would like to introduce you about Dr. Zakeel. Next. So our resource person is Mohamed Zakeel, Mohamed Cassim, Ph.D., or you can call him Dr. Zakeel. He is a research scientist from Commonwealth Scientific and Industrial Research Organization, or we known as CSIRO in Canberra, Australia. So his background, Dr. Zakeel received his honors in Bachelor of Science in Agriculture in 2006, and he continued his master degrees in biotechnology in Sri Lanka, and he received his master in 2014. Dr. Zakeel graduated from his doctoral degree at the University of Queensland, Brisbane, 2022. He is expertise on the plant pathology, especially on the molecular biology and epidemiology and many things. Because I think we hope, Zakeel, you can share your expertise and about epidemiology and molecular epidemiology with our students now. The students are from phytopathology. They are master students. So I think you can do a presentation for 1 hour and we can continue with discussion. Okay, time is yours.
Thank you. So thank you, everyone. Thanks, Ita, for inviting me for this guest lecture. Please feel free to stop anywhere if you don't understand any of the things. Let me share the screen here. Couldn't find it. Share. Yep. Can you see my screen?
Yes.
All right, thank you. Go to the, yep. So yeah, so as Itas mentioned, I will talk a bit about plant disease epidemiology, and I'll bring some examples from case studies, particularly studies which I carried out during my PhD and through some collaborations now and then, and also a bit of modeling of pathogen spread across landscapes. So first of all, let me give you some definitions about epidemiology. Hope you may have studied about it a bit, but anyway, for the sake of doing a course on epidemiology or doing a guest lecture on epidemiology, I thought it's better to give you some basic definitions of epidemiology. The term epidemiology has two components. Give me a second. I'm getting a.
Sorry for interrupting you.
Yeah.
Could you please your PPT?
I think it's slideshow now. You don't see it?
Not yet.
Why is that? Stop sharing. 1 minute. Sorry for that. Share screen, Windows, epidemiology. Now you can see the slides,
Yes, we can see.
right?
Could you please?
N-
Yes, that's it.
Is it slide share mode or now only the?
Yes, please click that button.
Yeah. Now you can see?
Wait a minute. I think it's still loading.
Is it?
Kayaknya itu deh pilihan share screen-nya.
Okay, it's all right. You can continue now.
You can't see. You can see it or you can't see it.
We can see, but not in a slideshow mode.
Oh, why is that? Wait, give me a second. How about it now?
Still not changing.
Oh, slideshow. Can you see it now? Slideshow mode or
No.
not it?
Yeah. No.
Why is that?
Itu kayak boleh bantu. Hello sir, may I help you for the
Yep.
share screen? Okay,
Yep. Yep.
you can choose the share screen mode,
Yes.
and then
1 minute. Yeah.
I'm sorry, you first end the share screen first.
Okay. Stop share. Yep.
Yeah, and then go share screen again.
Share screen. Yep.
Yeah. Is there any option to select the
Yeah. So
slideshow
I have
mode?
to select. I have to select Windows, and there are a couple of window, and then I have to click on the one that I wanted to share, the PowerPoint.
Yeah, the slideshow one, not the home PowerPoint view, the slideshow PowerPoint.
Sorry, I didn't get you.
Yeah. Yeah, in the share
Can you?
screen mode,
Yeah.
there is two option for PowerPoint. The one is on slideshow mode, and the other one is for the home PowerPoint. You choose the slideshow mode.
Wait 1 minute. Yep. Can you see it now?
No, the slideshow is still on home, not the slideshow mode.
Okay. It didn't give me two options.
Okay.
It didn't give me two options. Anyway, what I will do is, give me a
Okay.
second. I will.
Can you open the PowerPoint first, and then click the slideshow mode?
Yeah.
Okay.
Then
Is it on slideshow right now?
now, so yeah.
Okay. Back to the Zoom.
Hang on. How can I go there?
You can.
Because I'm currently using one single screen. I just came to a meeting room.
You can press the keyboard Alt and Tab. Press the Alt
Alt, yeah.
and Tab to choose the window.
Tap the windows. Yeah. No, it doesn't work.
Okay.
Nope. Why is that? All right. So what I can do is, the other option is, I can link to the screen here. Give me a second.
Okay.
Yeah, that's another option. We'll try that one. Yep. So where's this one? Let's go here. Share screen. Windows. Yep. Can you see it now?
Still on the. Okay, wait. Can you press the display setting, maybe?
Yeah, there should be some issues with display setting, I guess. Yeah. Yeah, let me go to that. Settings. Display setting. Yep. Then so I have to choose single monitor. Maybe because I have multiple monitor. Oh, sorry.
You have multiple monitor.
Yeah,
There is
yeah, yeah.
two screen, right?
Yeah, yeah, yeah.
Okay. If you stop share and then you click share screen again.
1 minute. Stop sharing.
Okay.
And share screen again. Yeah.
Okay. Is the second monitor showing your
Yes,
slideshow?
second. Yeah, yeah, yeah. I have to
Okay.
click that one, I think.
Yeah. Okay.
Now you can see full screen. Okay. Yeah.
Yes. Okay. Sorry everyone for like a little bit trouble, but yeah, we can
Yeah,
start with this.
that's fine.
Like, I'm sorry. Yeah.
That's okay. No problem. Yeah, yeah, yeah.
Okay.
Anyway, that's sorted out. Yeah, that's fine.
Okay.
Yeah.
Okay.
So yeah, let's continue from here. And yeah, the term epidemiology has two components: epidemic plus logy. As you may know, logy is science or study. So we can say epidemiology is the study of epidemic or a science of epidemic. And again, the term epidemic is derived from a Greek word epidemios, and the epidemios has two parts, epi and demos. Epi is on or upon. For example, you may have heard about a word, I mean, like epiphytes, plants growing on the surface of plants. So epi means on or upon, on the surface. And demos means people. So that's where the word demography comes from. So epidemios means what is among people. So because epidemiology is a field of study which was originally developed to study the diseases among people. So the epidemic is also known as epiphytotic. This word epiphytotic has been used long ago. I mean, yeah, it was used by Unger in 1830s and even in 1920s. This epiphytotic was used instead of epidemic. So the field epidemiology can also be known as epiphytotheology. Look at the word epiphytotheology. It is not epiphytology. So epiphytology is something else. It's study or science of epiphytes. So, but the term epidemic has been used for plants for a long time. For example, Duhamel has used it in 1728, and other peoples have used in 1850s and so on. So therefore, there is no valid reason for us to use still the word epiphytotheology instead of epidemiology. So therefore, currently most of the people use the word only epidemiology instead of epiphytotheology. Hope this makes sense. So epidemiology, as I already mentioned, is a study of the change in disease intensity and maybe spread in a host population over time and space. So in this definition, I have highlighted a few, or rather given a few keywords in different color. So one is change, disease, host, population, time, and space. Uh, all right, I'll just take a pointer. Yeah. So the first thing is, first word is change. So as you know, change is a dynamic process. So mostly in epidemiology, it's a process of increase. And the disease is a second keyword. So that means it is not just the pathogen or crop or the plant, but all aspect of the disease, which means the host plant, pathogen, and also the environmental interactions. So everything relating to this uh, disease onset and the progress are important when it comes to epidemiology. And the third aspect is host. So that is the infected or potentially infected organisms by another organisms, that is the pathogen. And then the population of host, and also the population of pathogen. And also if the disease is transmitted by a vector-borne diseases, then again, a vector population is also important. So it's a population of host and the population of the causal agent, the pathogen, maybe a bacteria or fungi or virus or phytoplasma and so on. And also if the pathogen is transmitted by a vector, for example, an insect vector, then the vector population is also important in epidemiology. Then the other aspects are time and space. These are physical dimensions of interest. Sorry. Now let's look at some history. When this field originated, how it was, used and, progressed to date. If we look at epidemic, the first use of the term epidemic was by Hippocrates. So in 400 BC, yeah, around 400 BC. And then around 340 BC, Theophrastus used this term and he worked on, he discussed about plant diseases in field and the environmental influences on plant diseases. And then, various other people like Ernst Albert Gorman in 1946, he talked about the principles of plant infection and infection cycle. So maybe the disease cycle, something, more or less related to disease cycle. We'll discuss about it. Yeah. And people like Large in 1952 talked about disease progress curve and disease assessment. And then Horsfall in 1960s talked about spore dispersal and prediction of diseases. And then Van der Planck in 1963, he, showed some models, developed some models for disease and established the science of plant disease epidemiology. Now we will look at the key elements of key elements of an epidemic. If you look at these elements, there are three key elements, as I already mentioned to you in the slide before. The host, pathogen, and environment are the key elements of an epidemic. For a disease to start and progress, these key elements should be satisfied. For example, so when it comes to pathogen, so if you want to assess the amount of disease in terms of epidemiology, so you may have in your pathology lectures, you may have studied about this disease cycle, disease triangle. So there should be a host, which should be susceptible. There should be a pathogen, which should be a virulent, and an environment should be conducive for the disease to develop. So for us to, estimate the disease, so we focus on the pathogen factors, for example, total amount of virulence the pathogen has, the abundance of the pathogen, so how much pathogens are there in the field, and so on. And also when it comes to the host factors, we need the total of the susceptibility. What proportion of your field is with susceptible host? what proportion of the field is with resistant host, how much susceptible, whether it is moderately susceptible or highly susceptible or resistant. So the scale is also important. And then the environment, the total of the conditions of the environment that are favorable for the disease to develop. And in addition to these three elements, there is another element that is also important, that is the time factor. So time is also important. So maybe at the beginning, of an infection event, the disease intensity or severity may be very less, and after a while, maybe it may be higher. And also, it does not mean the time from infection to the end, but also the time of infection of the host. I mean, which time of the host the infection occurs, for example, whether it is seedling stage or a growth stage or reproductive stage and so on. And then, so if you consider these four elements, then we call this as disease tetrahedron. This is triangle, and the third one is disease pyramid, where we consider one more factor in addition to those four, that is human or anthropogenic factor. So an epidemic consists of, as we have discussed, a population of infected individuals, that is the host individuals, infected host population, in a host population, and the change in diseased individual over time and space. So this captures all those key words here. So in an epidemic, we consider a population of infected individuals in a host population and its change over time and space. So if you have any questions, you can stop me anytime and ask them. I mean, yeah. But anyway, in between, I will give some time for us to stop and think about it and ask, get your questions clarified. If you look at the plant factors that affect the development of epidemics, there are a few factors that affect. The first one is host genetic resistance. So there can be two types of resistance, vertical and horizontal resistance. If you look at the vertical resistance, that is the gene for gene relationship. So in plants, we have, strain specific resistance, for example, for a particular strain of a pathogen, there can be genes, resistant genes to confer resistance. So that is we call vertical resistance. The second type of resistance is horizontal resistance, which is broad spectrum and which is not strain specific. That is the resistance for a variety of pathogens and strains and so on, which is conferred by thick cell wall or cuticle or having hairs or hairy structures on the leaf and so on. Okay. So that is known as horizontal resistance. And the second factor is the degree of host genetic uniformity in the field. That is whether you have a monoculture in the field, for example, wheat or rice or soybean or canola or maize only in your field. So that's monoculture. So you keep on growing only one crop. Or whether it is a mixture. So we have, two different or three different varieties in a mixture. For example, you have maize with, say, for example, a pulse, a vegetable with another veggie crop or something like that. Or whether you have a mosaic structure in your landscape, for example, say in your one field is say wheat here and canola here, soybean here, and so on. It's a mosaic structure on a landscape. The third factor is the crop type, whether your crop is an annual, whether it is a foliar or fruit disease. If they are foliar fruit diseases that they develop very rapidly, whether it is a perennial, for example, woody perennial plants take longer time for disease to develop, to come to an epidemic level. And then plant age. This is also an important factor because some plants are susceptible during early growth stages and they become resistant when they are mature. So for example, when some plants become mature, so they have some genes expressed when they become mature, for example, APR, adult plant resistance. So these are four important plant factors that affect the development of an epidemic. Now let's look at the pathogen factors that affect the development of an epidemic. The first one is the level of virulence. So this determines the rate of inoculum production, how much of inoculum is produced at how fast. For example, if the pathogen is highly virulent, so you'll produce a lot of profused amount of inoculum within a short period of time. So that will eventually lead to a high level of epidemic. And then the reproduction rate of the pathogen, sorry, reproduction type of the pathogen. So the reproduction rate comes under the level of virulence. The reproduction type, whether the pathogen is monocyclic, polycyclic, or polyetic. Monocyclic means, I will show a diagram of the cycle in the next slide. Monocyclic means the infection occurs and the plant become diseased and the plant dies, and then the pathogen undergo overwintering, or we call overseasoning stage, and then infect, produce inoculum for the next season and infect the crop in the next season. So one disease cycle per cropping season. So the example for these monocyclic pathogens are most of the soil-borne diseases, for example, Fusarium wilt. And then polycyclic pathogen, which is very detrimental really, which causes sudden and catastrophic epidemic, and they are mostly airborne. Here what happened is that, I will just show the next slide so that it'll be easy for you to understand. So this is the monocyclic cycle. So the primary inoculum infect the plant, and the primary infection happens, and then plant dies or recovers, and the spores are produced, and spores, survive the unfavorable condition. So we call overwintering or overseasoning, and produce primary inoculum for the next season. So this is the cycle, and most of the time, this is the time during which, for the pathogen which can produce sexual spores, sexual cycle happens during this time. And then when it comes to polycyclic pathogens, the primary inoculum produce the infection, cause the infection, and then the infected plants. And then from the infected plant, what happens? The secondary cycle happens. I mean, the infected plants, from the infected plants, the pathogen produce asexual spores, and and infect. The secondary infection happens, infect, host tissues, and then the tissues become dead, and then again on the dead tissues, the spores are produced, and they keep on, repeatedly this cycle happens, and when the season ends, so then the pathogen undergo overwintering, and start the next cycle next season. So the per season, so it produce a lot of disease cycle. So that is why we call it as polycyclic. For example, black leg in canola, and ascochyta in um, chickpea. So this is an Ascochyta symptom in chickpea, and this is a pot infection in by Ascochyta rabiei in chickpea. Um, yeah. Then a polyetic. The polyetic reproduction, it can be by a monoculture or in perennial in plants in tropics. That can be caused by both monocyclic and polycyclic pathogens. Here, what happens is that uh, the infection happens and they produce inoculum over and over and over again over a period of time, maybe 2 years, 3 years, 4 years, and then lead to an epidemic. So it is mostly possible in perennial plants in tropics or in monoculture. For example, if you grow one single crop over and over, I mean, repeatedly for several years. For example, you grow rice this year, rice next year, rice third year. So what happens, the inoculum built up in this year, and again it produce some, give some, carry some inoculum to the next year, and again it produce more inoculum, and it keeps on developing to a level where it becomes epidemic. The third factor is quantity of inoculum near the host, how much of inoculum that we get near the host. So for example, if your neighbor has a lot of inoculum, source of inoculum, then the chance of getting, uh, the disease for you is very high. Then pathogen ecology, whether the pathogen reproduces on the surface of plant or within the plant, inside the plant tissues, or on infected plant parts in the soil, for example, roots, or on crop residues, for example, stubble, and so on. Then the final factor is the dispersal mode, how it is transmitted from one place to the other, whether it is by strong winds and breeze, so which mostly cause very sudden widespread epidemics, or whether it is by airborne vectors or just wind-blown, rain splashed, or by planting materials through seeds, tubers, bulbs, etc., or by vectors such as insects or beetles or animals, or whether it is soil-borne. If it is soil-borne, maybe by anthropogenic activities. For example, if a person who visited one farm which has a soil-borne pathogen can carry the pathogen on his boot, yeah. So these vector-borne, wind-blown, or rain-splashed, or the disease transmitted via planting material, or vector-borne diseases, or soil-borne diseases, they are usually local epidemic, and they are slow spreading, as opposed to strong wind or breeze, borne dispersal mechanisms. This is something we have already talked about. And then environmental factors affecting the development of an epidemic. There are two key factors. They are temperature and moisture. When it comes to moisture, variety of components of moisture, for example, rainfall, or it may be a humidity, dew, and etc. So this factor is important. It's an important factor for diseases caused by oomycetes, fungi, bacteria, and nematodes, but not much important for viral diseases or phytoplasma diseases. And then time, the duration and frequency of rainfall, favorable temperature, the time at which you get favorable temperature, the time at which you find the presence or the prevalence of vectors in abundance, or the season of the year. Some seasons, you get more epidemic or disease, particularly in tropical countries, because in sometimes you get in tropics you get high humid conditions which may favor fungal diseases and so on. And then human factors, anthropogenic factors, for example, factors like selection of sites, land preparation, and planting material selection, whether you select resistant cultivars or susceptible cultivars, and cultural practices that you adopt, and disease management measures that you implement, and also the introduction of new pathogens. For example, exotic pathogens. You bring knowingly or unknowingly, unintentionally, some pathogens from out of your country. So even though you have some, strict quarantine measures, can be ways in which it can, come, you know, by human activities. All right. So before moving on to the next topic, which is about disease progress curve and estimation of disease, any questions? Or shall I continue?
If you have any questions, please feel free to ask. You can just interrupt him.
Yeah.
It's okay for doing interruption during his lecture, so feel free. Okay,
Yeah.
Zakeel, please, you can continue.
Yes. Okay. So if you have any question, just put your hand up and stop me so I can. Yeah. All right. So next, let's talk about disease progress curve, which is something you always come across in, epidemiology. This is the representation of plant disease over time. So, which is mostly shown by a graph disease intensity, which is plotted in the y-axis over time in the x-axis. So this is generally used to predict future disease development, how the disease progress, and also to assess disease management strategies. We look at the disease progress curve for one management strategy against the other and see which one is, doing better job. And also we use this to compare the effects of environmental factors on disease development. And then we use disease progress curve to forecast disease for effective control. So what time, how it is moving, whether it is on a declining. For example, if you look at here, for example, you don't need to apply any control measures here. This is because very slow progress, as opposed to something like this here. See, and to evaluate the level of resistance in improved varieties. Suppose you get a couple of improved varieties and some, moderately resistant varieties and susceptible lines. Now you want to check whether your improved variety is really doing a better job. What you can do is you can, so artificially inoculate, the plants and then, develop a disease progress curve and see whether really your improved varieties are, there's a typo here, improved here, varieties are really doing a better job. These are some examples of disease progress curve. In the x-axis, you can use any parameter to represent disease intensity. For example, here, they have used, I have got this from Roop and others in 1982, work on maize gray leaf spot, where they have used lesions per leaf. How many lesions? So yeah, per leaf. And some people use disease incidence in percentage. For example, here, Howard and others in 1992 on Phytophthora blight of pepper seedlings. And then in Fusarium kernel rot of maize, they have used percent kernel infected. Or we can use even, yeah, percent kernel infected again here in this paper. So this is a typical monocyclic pathogen. So you get a curve like this. And if you plot this one in a Lana function, so you get a straight line. And let's talk about how this curve behave for a polycyclic, a typical polycyclic pathogens. Now you know what monocyclic and polycyclic pathogens are. So if you look at a disease progress curve for a typical polycyclic pathogen, now I will explain this with an example here. So suppose you have a field which has 4 million plants, potato plants. 4 million potato plants mean 4 into 10 to the power 6 plants. And you get an epidemic starting with a single lesion which causes potato late blight, right? One single lesion. The source of this lesion could be spore produced on an infected tuber within this field. You may have brought one just single tuber with infection, or maybe a spore arrived from the next door, the near field. The spore just jumped and caused infection here. Just single plant infected. And on this plant, how much infection or the lesion covers the total plant? So one late blight lesion covers 0.1% of the foliage. That means if you take one plant, 0.001 of the plant is infected, or rather of the part is covered with the lesion. Okay, so that means 1,000th of a plant. If you divide that plant into 1,000, only one part is covered by the affected tissue, right? But if you look at the relationship between the number of lesions and the foliage covered by the lesion, depends on two main factors. One is pathogen strain, whether it is a aggressive strain or not. If the strain is very aggressive, so it produces larger lesion. And then the cultivar, the plant cultivar. For example, if the plant cultivar is susceptible, then again the lesion size become larger. A larger leaf area will be covered by the lesion. Right? Now let's calculate disease intensity. At time 0, so disease intensity is the proportion of foliage in the field covered by the lesion. Now, you know, in one plant, it covers only 1,000th of a plant. That means 0.001. And you have how many plants? 4 million plants. So in the field, so it is just very small fraction. So if you want to find the disease intensity at the first initial time t equal to 0, that's the first infection. So that will be 0.001 into how many plants in your field? 4 into 10 to the power 6. That means 2.5 into 10 to the power minus 10. So you have to divide this by this many plants if you want to find the disease intensity for the field. So it will be 2.5 into 10 to the power minus 10. That is at time t0. Suppose if the number of lesions double every 2 days. So you have one lesion today, day after tomorrow, it becomes 2. So in 4 days, it becomes 4. So in the 6th day, it will be 8. It keeps on increasing like that exponentially. So there will be 4 into 10 to the power 6 on average, 4 into 10 to the power 6 lesions in about 44 days. You can calculate. So at t equal to 44 days, the intensity will be, so you have to multiply this by how much? This many lesions, 4 into 10 to the power 6. So that will be, y will be equal to 0.001. If you keep on doing this for several time points, like for example, 64, 77, 70, 77, 88, it keeps on increasing. But what happens? Initially, it will increase slowly and then starts exponentially and then starts to increase at a decreasing rate. That is because of the slowing effect of loss of disease-free foliage. Because what happened at the beginning, one becomes 2, 2 becomes 4, 4 becomes 8. Then after some times, for example, 44 days. So 44 days, it will be 4 into 10 to the power 6. Then it becomes double here. So it will increase exponentially for some times. Then what happens? Most of your healthy tissues become infected, and there will be depletion or loss of disease-free foliage, healthy foliage for your spores to land and cause infection. So therefore, what happens? It will start, stabilizing, at towards 100%. So that is why it keeps on, it shows this trend. Hope this is clear. So for polycyclic pathogens, you get a graph like this, and if you transform into, if you do a log transformation for the disease intensity, which will produce a linear model. So for example, you can develop a model for polycyclic epidemic, something like this, where X0, X0 is the initial inoculum, R is the rate of disease increase, and time is the time. Yes. Now let's talk about how we measure plant disease. So measuring plant diseases. So the measuring of plant disease is the disease assessment, or some people call it as disease estimation. So there are various measures or estimation methods depending on pathosystems. So some are qualitative, some are quantitative, and some are sort of both. And the disease estimation is done per plant or per unit. It may be a leaf, a unit may be a branch of a plant, and so on. First such measure or the estimation is disease incidence. So disease incidence is the actual number or the proportion of diseased plants out of the total number of plants observed. For example, if you have 1,000 plants and if you get, say, 500 infected, so the disease incidence is 500 out of 1,000. That means 0.5. And again, depending on, if you are looking at plant unit, for example, you are looking at leaf as your unit to assess your disease, then we call leaf disease incidence. That is the proportion of diseased leaves. If your unit is a whole plant, then plant disease incidence. That is the proportion of diseased plants. For example, you have a field which has 2,000 plants. 1,000 plants in one side are healthy. Another 1,000 plants are infected. And each of these infected 1,000 plants has half of the plant is covered by lesion. 50% of the plant is covered by lesion. So this is half of the field, this is half of the field, right? So the incidence, how much is the incidence? That is 0.5. Why? There are 2,000 plants. Out of 2,000 plants, 1,000 is infected or diseased. So 1,000 divided by 2,000, that is 0.5 or half. Then disease prevalence. That is the proportion or number of field with diseased plants. So in this case, the prevalence is half because we have 1 out of 2 fields. Suppose you have 4 fields. So similar to this, there are 4 fields here. Say these 3 fields, there are 2 fields here, hypothetical fields. 1, 2, 3. These 3 fields are not infected, healthy. Only this one is infected. Then the prevalence is 1 upon 4.25 because only one field, regardless of how many plants. It doesn't necessarily need to have all the plant in this field is infected. Here I already mentioned this is a single field, but for this hypothesis, hypothetically consider this is a separate field, this is separate, and there are 2 more fields. Okay. So suppose there are 5 fields, 5 farms, and you have 2 farms with infected plants. Not all the plants are infected, only a few, and in this farm 2, in this farm 3, and all the other farms have no infection, then still the prevalence is 2 upon 3. So it's all about the farms or the fields. Then the disease severity, which is very important when it comes to epidemiology. This is the relative or absolute area of plant tissues affected by disease, which is important because that is something related to the yield. So in this case, the mean disease severity can be calculated by, so you know, the incidence is, because this is the relative or absolute area of the plant tissues affected by disease. Now, you know, half of the plants are affected here. And out of those half in this field, each plant has only 50% lesion. That means 0.5 per plant has lesion. So in this field, the entire field, this whole area, the mean disease severity is half of the field is affected, and in each, or half of the, not field, half of the plants, out of 2,000, 1,000 plants are affected. That means 0.5 plants. And again, in each plant, how much of the area of a plant is affected? 0.5, 50%. So that will be 0.5 multiplied by 0.5, that is 0.25. That is mean disease severity. Does it make sense? Okay, I hope yes. Then further on disease severity, for many diseases, it is the area of plant surface covered by lesions and the disease area of an individual plant or mean disease area of a sample. This is mostly measured using assessment scales or by determining the area under disease progress curve, which we already discussed. Then severity. The disease severity is mostly preferred for foliar diseases. So because of the continuous variable, because there can be very less severe, highly moderate, or we can have a scale, for example, to measure disease severity, and which is relatively easy to visualize because foliar diseases are quite easy to visualize them. And there are various methods to assess the severity of foliar diseases. Whereas for viral diseases or root diseases such as wilting and yellowing, so the preferred method of disease estimation is incidence. So how many out of, maybe in the fields, maybe 1,000 or per quartet. Okay, so per quartet you have this many plants. So how many of them are, infected or diseased and so on. Because it is very hard or difficult to define the severity when it comes to viral diseases or root diseases. Sometimes we may need to do sort of, molecular diagnostics to see whether a plant has disease. So we can't visualize, we can use visual symptoms to characterize the severity in viral diseases or root diseases. Then the next assessment method is yield loss, which is the proportion of yield loss due to the disease. This results in economic loss. And then another important thing to consider here is the disease gradient, the change in disease severity along straight line away from the source of inoculum. Suppose the inoculum is somewhere here. So if you move away from the inoculum source in a straight line, how the disease is, reducing. So that is we call disease gradient. I'll show some examples from some case studies in later slides. This indicates the dispersal potential of a pathogen or inoculum in a single disease cycle or from a single source. More on disease intensity or severity. The magnitude of disease intensity is not an indication of whether an epidemic has occurred or not. So the disease intensity can be low or high at a given time. That depends on the initial level of disease and the rate of increase. So if you want to define AUDPC, so area under disease progress curve, which is used to measure, disease severity, this is a quantitative measure of disease intensity over time, commonly used to compare disease progress among different treatments or environments, as we discussed. For this, if you want to calculate AUDPC for a situation, what you have to do is you have to follow these 3 steps. Number 1, you have to collect data. Gather disease severity data at regular intervals over time and record the percentage of disease severity or another appropriate measure at each time point. For example, some people use scales, 1 to 10 scale, to record the disease severity or percentage, whatever that suits you. And then the time intervals, you have to record it at a regular sort of time interval, and ensure that the time intervals between observations are consistent. If they are not, use the exact time difference in your calculations. So for example, here I have used, at some points we have used 3 and here 4 and so on. Yeah. And then the easiest way is to use a trapezoidal rule to calculate AUDPC. So that is, if this is your disease severity over time and this is your plot, so what you do is you just break into regular intervals, and then you produce some trapezoid, you see here, and then find the area of this trapezoid. So trapezoid is having 2 parallel sides, and then you calculate these areas and then add them together, and that produces your area under disease curve. All right. So I'll use this graph in the next slide to see how to calculate the AUDPC. This is time recorded, and this is the severity at different time points. Before moving to that example, I have to introduce one more definition, that is a word, a term, that is pandemic. So pandemic is an epidemic that occur over large areas with high level of disease. For example, COVID, that occurred in many countries at very large scale. So then only we call it as pandemic. Suppose if you get something like that at large scale, but only in one place or one country, then that is still epidemic. Now let's look at how to calculate this is AUDPC using this table. The value is given here. So we can use this formula, summation of those different values here. If you look at these trapezoidal, these 2. So how do you calculate the area of trapezoid? You add these parallel lines and take the half of it and multiply by this height, this one, the time. So that is what you have done here. You multiplied by time, and you have added these 2 sides and divided by 2, and summation of both all at different time points. So for the interval 1, so we have different intervals here. So this interval 0 to 4, 4 to 7, and so on. So for each interval, so we have added those times up and divided by 2, and then found this time difference and multiplied that time difference by these disease severity difference, that value, the mean of the disease severity, and then you have found this for each trapezoid. And then what you do, we add them together to produce AUDPC, which is 462.5. Otherwise, what you can do, you can just write a simple code. Suppose I did this yesterday. I wrote a small code. I built a small model, a logistic. I fit a logistic model to find severity. I use severity is equal to this model, and then found the model was significant, and then I found the, you know, the AUDPC, which is a bit higher than this because I fitted the model here, right? Because this is not accurate because there can be some, you know, because it's just sort of, because the other thing is it is not always, this graph is not always, you know, sort of straight line. That is where it becomes important to fit a model. So we can fit different models. Even we can fit a, not only a logistic, we can fit a linear model or any regression model, because the main reason for fitting a model to accurately calculate AUDPZ is because this curve may not be sort of, you know, regular shaped or straight line for you. So it's always best, or it's a good idea to fit a model and then calculate the, and see whether the model fits well, and then calculate the AUDPC. So now let's introduce some case studies where I have used some basic epidemiology, which I have discussed to you now. So during my PhD, I worked on a enigmatic disease called abnormal vertical growth. So we call AVG. The symptoms are excessive upright growth. This is a normal tree. You can see here, so upright growth, you know, it grows upright, which, you know, reduces yield significantly. And the impact is very severe, and the cause was unknown, even though the disease was increasing at an increasing rate. And previous studies suggested it was not pathogenic soil nematode, not due to soil nutrient, not soil physical chemical properties, not due to insects, not due to sunlight or other factors, but soil moisture was sort of triggering the symptom. So this, if you are interested, you can read this paper published in this journal, Phytopathology. So what I did was, I asked some questions like, can the pattern of AVG spread help us to, you know, shed light on its epidemiology? So then I collected historical data from 6 commercial orchards and surveyed in 2012 and 18 and analyzed for the distribution pattern of affected trees, whether they are, the affected trees are aggregated or they are just random. Because if it is random, so maybe the disease may be, you know, sort of vector disease. If it is aggregated, so if it's most unlikely a vector disease, whether it is moving from affected tree to healthy tree, how it is moving, whether it is at a fast rate, is changing incidence over time, is slow or rapidly. So what I did was I fitted the data to 2 models, binomial and beta binomial. So binomial is a distribution which shows that the affected trees are sort of random. So beta binomial, if your data fits well with a beta binomial distribution, that means your affected trees are sort of aggregated. They are available in small patches. So then we had to find, so what I did was I found the better fit using a chi-square goodness of fit comparison. And also I calculated Fisher's index of dispersion by, you know, dividing observed variance by the binomial variance. So this Fisher's index gives some, again, an idea of the distribution pattern. If your Fisher's index is less than 1, that shows a uniform distribution. The affected trees are uniformly distributed in the orchard. If your Fisher's index is equal to 0, it's sort of random. And if it is higher than, equal to 1, it is random. And if it is higher than 1, it is aggregated at cohort level. This is a table from that published paper. here you can see here. So in all of these, you know, I use different coated size, you know, different orchards, you know, and this is the observed incidence. Can you see here? So what percentage of the trees are infected out of this many trees? So totally we had 2,304 trees, 4.86%. In this case, out of 480, so 46, almost 50%, that means 240 or 220 trees are affected here. You see here, so that is the incidence in this year and this year, 2018 and 12. So this is in a small scale. So when you plot these, you know, affected individual trees in different orchards or, you know, in different time, we can see here, you know, these are sort of aggregated patterns here, you know, some trees are here. These are random, but mostly, you know, there are patterns of aggregation. So this result shows aggregated pattern. And also, if you look at from 2012 to 2018, there is an increase in number. And also the aggregated, you know, patches. And the increase, the rate of increase was 2% per year. So from these, we found that the spread was mainly between neighboring trees. You see here. So from here, it is moving next, and it moving, you know, along this, because this is the row, and this is between row, you know, this side, and the opposite trees in different rows also, you know, the spread occurred. That is why it is moving this side as well, and also this way. And this is another table from that paper where we can see these log likelihood ratio statistics to check the better fit of the beta binomial model here in this case than the binomial. I'm not going into detail about these are the results of the LRS. These are, you know, sort of negative values here in this one, the binomial. So the significant log likelihood ratio test statistics was tested for, you know, the p-value less than 0.001. That means 1%, yeah, even 0.1% significance level. And here we can see, you know, all of the beta binomial, you know, showed better fit. So I just calculated LRS for beta binomial fit as opposed to binomial. And also to map. So as I said, so this is an important and interesting way of, you know, expressing how the disease is spreading over time and space. And the second one is we can use heat maps to show the incidence, again, over time and space. So here I used, for these two graphs are from the same paper, from the same study. So how, you know, spatially, the incidence is, you know, sort of increasing. So for example, I have used a quadrant which covers 8 trees. So out of 8 trees, how many trees, you know, 0 is, that is here, these quarters have no infection. So all of them are healthy. So white indicates healthy trees. So light color, less, 1, 2, 2, 3, and it keeps on increasing, and the highest is 8. The darker color means all the 8 trees in this quadrant are diseased. Same like here, the quadrant side is 4. So these small, you know, darker, you know, scars indicates that all the 4 trees in that place are infected, whereas here, all of these are, you know, sort of healthy. And this is from a different study that I collaborated with on a spatial distribution of corn standing disease incidence caused by a viral and phytoplasma in Brazil. So we are currently working on a couple of manuscripts, and this was presented at a conference in Brazil this year. So that is one way. Another way of, you know, presenting your disease incidence using heat maps. And also we can use sort of frequency distributions of number of disease host or disease plants. So here in this case, what I have used is the frequency. I have plotted the frequency of the observed frequency and the how about the frequency of aggregated or random, because I just modeled it and I found these different parameters. Don't worry too much about it. Considering the time, I will just rush a bit. But yeah, this is another way to present. If you don't have these observed, sorry, random and aggregated, if you don't fit the your data to a model, at least you can just plot the observed frequencies. For example, this one says how many are in your quoted are with no disease is a large number. This is indicating 1. This is 2, 3, 4. Same like here. And the zero dark color is the observed one, the real one. And this one gray is the aggregated. Whether this fits with aggregated or random, that's the main goal here. So in most of these cases, my one is going with the gray one here. If you look at here, so gray is closer here. Gray is closer to the darker one. So that is why my data is fitting more to the aggregated than random distribution. And if you are interested in looking at larger scale, so there are a couple of tests that you can do. That one is you can develop some graphs like this, or you can do a sort of semi-variogram. So we call this a semi-variogram. And this in this semi-variogram, this is we call the range. If the range is large, that means the clustering of affected trees happens. And yeah, and here also at a larger scale, we can see sort of aggregated patterns here. Yep. This is done through an analysis called SADIE, S-A-D-I-E. If you're interested, you can just read some more or read my article. You can find more information. From this work, what we found was ABG distribution is aggregated. So the nature of spread is aggregated, clustered, and the rate of spread was slow. So from this, we formulated some hypothesis that this disease was not caused by organisms that produce large amount of wind-borne or spores or inoculum, and also not spread by highly mobile insect vectors such as aphids, which are vectors of virus, or leafhoppers, which are mostly the vectors of phytoplasma. So it is very unlikely that this is caused by phytoplasmas of viruses. But later we proved that they are not caused by any of these. And also they move mostly along the rows. So most likely we found that they are transmitted by root grafting or root to root direct contacts. So that's about the basics of the epidemics and epidemiology, epidemic dynamics. Now we'll talk a little bit about modeling. So when it comes to epidemiology, now everyone talks about modeling of epidemiology, epidemic dynamics. So the basics of models, when you take the term model, so the model can be a mental model or tangible model. So mental models may be theories or the hypothesis that we think of, or conceptual frameworks, or tangible models. So tangible models can be of two types, physical and abstract models. The physical models can be scale models or prototypes or mock-ups and so on. For example, you develop an aircraft. So you develop a prototype on the aircraft, so which is a physical model. Then in abstract model, there can be two types, a qualitative and quantitative model. Qualitative model, for example, flowchart, conceptual frameworks or maps, and logical frameworks or theoretical models and so on. Then quantitative models, it can be either statistical model, mathematical model, computational model. Now we use computational model for the AI, and machine learning models, econometric model, and so on. And these models can be stochastic. it may change, it has a changing, component. So more about these models, some description about these models and some examples. So for example, mental model. These are cognitive constructs that individuals use to understand and interact with the world. They are not physical models, but exist in the mind and help in predicting outcomes and making decisions. For example, a researcher thinks and hypothesizes that a particular plant disease spreads faster in humid condition. So then he can develop a tangible model from that. For example, tangible models these are models that have a physical presence or are directly measurable and observable. For example, a physical model. So I said tangible model can be two types. A physical model. These models represent real world objects or systems with physical components that can be touched or manipulated. For example, from the previous example, the plant disease spread faster in humid condition. So we can have a greenhouse setup replicating various humidity level and observe whether the disease spread in different control environments differ from one to the other. Then abstract model is a uh, uh, is a model that is not physical but represents systems or concepts through symbolic or mathematical means. So as I said, this can be of two types, qualitative model and quantitative model. The qualitative model is a model that describes the characteristics or behaviors and relationship of the system or concept without using numerical measurements. For example, if you create a flow chart, say from this, that happened, from that, two things happens, and so on. Okay, so even that is very important when it comes to modeling, uh, epidemic dynamics. Then we have quantitative models. These models use numerical data and mathematical equations to represent the systems or concepts, allowing for precise measurement and prediction. For example, a mathematical model using differential equation to predict disease spread rate based on humidity level and various other factors, infection rate, inoculum density, wind speed, and so on, right? Now let's talk a bit about epidemiological modeling approaches. So there can be two approaches. One is data-driven approach or data-based approach. For example, we can use a linear regression. So something similar to the thing that I mentioned before to calculate AUDPC. So if you have some data, so you can, feed that data to a model and develop a model, test the model, and use that model to predict the AUDPC for different situations or scenarios. The second one is process-based approach. So that can be of two types, simulation models and predictive models. So examples for simulations models, for example, epid is a well-established model, which is based on two concepts, SIR, HLIR, we will discuss later more about it, or wheat pest model. And there are so many, simulation models available. And this model is a crop growth model, but that accounts for the effect of disease as well. Or predictive model. The predictive model is a model that predicts the effects of weather, host resistance, and inoculum pressure on the green leaf area or yield loss. Or the output can be AUDPC. So for these models, as I said here, these models are compartmental models. There are different types of compartmental models or different model types. The first one is SIR, or some people call HIR. That means, so if you consider the host is moving into different compartments, this has three compartments, SIR. That means susceptible, infected, and removed, or dead. So some people, instead of susceptible, they call healthy. So the So the removed, some people they call recovered. So because it depends. Sometimes when the disease happen, the plant will die and die forever. Sometimes when you spray fungicide, they are recovered, or due to their immunity, they recover and become healthy. But we consider it as removed from the system when you do modeling. The second is the model that has 4 compartments. So one additional compartment in addition to SIR or HIR, that is E, that is uh, exposed, or some people call latent. So first compartment is healthy or susceptible host. Then the inoculum comes, the hosts are exposed, infection starts, but it undergoes, they don't show, the host plant don't show symptoms. They undergo the latent period. So that is we call exposed or latent compartment. And then it shows the symptoms, or the host becomes infectious. That means the plant is infected and they now produce, they are infectious. That means they can produce inoculum to spread the disease. And then they are removed from the system. Maybe they are recovered, or maybe they are deceased or dead. So if you look at a graph, so how it is moving, for example, the healthy host start getting infected and it start declining. So the yellow shows the susceptible host, how it is happening. It's going down, whereas the maroon bar shows the infectious, start infecting, increasing, and then getting recovered. Whereas this one is the recovered one, it increasing. So at any time point, if you add all these values of this graph, it should be equal to 1 or 100%. So let's look in a bit of detail about the SIR model: susceptible, infectious or infected, and recovered or dead, or even immune to disease model. So from susceptible or healthy compartment to infected compartment for the disease to move. So we can, this susceptible, if you take a population of hosts, the number of susceptible hosts and number of infected hosts and number of recovered hosts will be equal to the total number of host plant in your field. And small s is the fraction of this susceptible. Small I is the fraction of infected. That means infected, number of infected divided by total host. And small r is the fraction of the recovered. And if you add all these fractions, it will be equal to 1. So we can use differential functions or the derivatives to find the changes from one compartment to the other. For example, ds by dt, that's a differential of the change from susceptible compartment to the infected compartment, is the rate at which healthy individuals become infected. The second one is the infected compartment. The rate, this is calculated based on di by dt, is the rate at which healthy individual become infected minus, we have to remove, because some of the infected one at that time point become recovered. So we have to subtract that, the rate at which infected individual reach the end of the infectious period. So then only we can calculate this part, the deceased. And then how much is recovered? How do you calculate? So this one gives the change in this compartment. This differential functions gives you the rate at which infected individual reach the end of the infectious period. So always we use this with a negative value because this is, declining. This number is, so, reduced in the healthy compartment, and this is the one which are infected, and this is the one which are, so recovered. So to calculate this, So this is your, so we calculate this change, the rate using this formula minus beta into S is the fraction of susceptible and I is fraction of infected. Because we need to know how much susceptible they are and how much infected they are. Then from this infected, again, some of the hosts become infected. So because this infected one produce the spores, so these two, the numbers, these fractions are very important. So we multiply this by beta, and this beta depends on three factors: alpha, theta, and psi. So alpha, because this rate is, I'll show you, that is the beta is the per capita transmission rate of disease. So that depends on three components. The first one is average number of spores each infectious individual produce per unit time. For example, this infectious host, so each of these produce how many spores? Because that is important to find this. And then the probability that an infectious units come into contact, because suppose if one plant, infected plant, produce 1,000 spores, not all those 1,000 spores will come and land on a plant surface. Some may go and land on a field, on the soil, maybe on some structures elsewhere. Some may not fall here, some outside the field. So what's the probability of an infectious unit come in contact with a healthy individual tissue? So that is also important. And again, the third one is what's the probability of that, the spores that are landing on the host, healthy host, will infect. Not all these, say suppose out of 1,000, 10 spores are landing on a healthy tissue. Out of those 10, only one spore will infect, or none will infect, or 0.01% is the probability of infection. So those three are important to determine this beta here. And that is important to determine how much from susceptible is going to be infected. Okay. So similarly, so here mu is important. That's the, here the mu is important to calculate how much. So that depends on how much, is removed or recovered, and this, that's a model with mu. We can calculate mu. Mu is the removal rate of infected plants, and always this removal rate of infected plants has a relationship between the infectious period or contagious period. So if you put 1 upon mu is always the average number of days that a plant stays contagious. That is the infectious period, we call, in other words. And then there is another important factor we call R0, R naught, that is basic reproductive number. That is the expected number of plants infected from an infected plants over the length of their sporulation period in a fully susceptible population. This is calculated by multiplying the expected number of close contacts per day. So how many spores are really landing on susceptible host per day, multiplied by average number of days that spores remain infectious, or the plant, sorry, not spores, the infectious plant stays infectious. That is, this is 1 upon mu. That is your, this value, 1 upon mu, average number of the infectious period. And multiplied by, this is your expected number of close contact per day. That's your per capita transmission rate of disease, beta. So if you multiply this, that gives you R0. So that is beta divided by mu. And then the second model is, we have one more compartment, healthy. From healthy, infection happens, and then host move into latent period. Then at the end of latent period, it moves into the infectious compartment, then removed compartment. So we have, in addition to this, we have one more here factor, that is omega. And if we calculate this, we can, just model, find out, how much. So I'm not going more, uh, uh, you know, into detail about how we calculate all those things. We can find some literature on this. So I'm currently working on a slightly modified model here. So now the spores are produced from infectious plants and as well as also from removed plants. So since I'm working on stubble-borne pathogens, the infection, the spores contribute for the cycle when it comes to polycyclic or diseases or stubble-borne diseases. In other words, or in addition to stubble-borne diseases, there can be spores on the stubble contributing to a new infection. Okay, so for these, as I said, so we have these changes in the from infection, from the healthy to latent period, and from latent period to healthy, change. So we can calculate the rate at which healthy individuals become infected minus rate at which latently infected individual become infectious. That is the latent period time. So that we can calculate by multiplying omega by latent period, um, the population in the latent period, and then this one by subtracting this the removed population from this component, and so on. Then before closing, I will concluding, I will just talk a bit about tracking of pathogens. There are various methods in our previous, experiments, and all of these experiments we use different tracking methods, particularly when it comes to fungal diseases. We use spore traps. Now there are well advanced spore traps available. There is one we call uh, there is a spore trap which is used to predict DNA. So they call prediction DNA spore trap, which is currently being developed by Berkhand and Rothamsted Research and Helix Farm in the UK. In this technique, in this trap, what they do is these traps collect spores, and the spore DNA is chemically released, and the quantitative fluorescent signal data is produced, and that is uh, sent for backup reporting of cough infection risk. And also, they are currently developing some molecular assays based on qPCR so that they can automate it, and they give, once they get this quantitative fluorescent signal, they report it, and they just give a sort of signal, say, okay, hi guys, you have this epidemic onset, or it's very high level or low level, things like that. And they are currently working on developing some uh, diagnostic assay which they can combine with these traps so that they can do sort of in-field assays and get this reported done as quick as possible. And then in Australia, there is a company that has developed some traps called BioScout, which is an automated airborne disease tracking system, which is a next generation spore trapping. Here, what they do is there is an autonomous spore sampler and an automated microscopy, and the spores are sampled, and the microscopy is used to to capture the images of spores, and the data is uploaded to a cloud-based server which runs a sort of machine learning algorithm to automatically identify the microscopic images of the spores and classify the spores in real time and provide information to the growers. Okay, so these are the exposed at least this level and so on. And also in China, some researchers are developing some detection of fungal spores based on a microfluidic separation enrichment technique, which is a sort of technique where what happens, there is a sort of microfluidic separation enrichment apparatus which suck the spores, and then sort of this is your microfluidic chip is there. Then they have an alternate current, and they detect the AC impedance, the resistance characteristics or impedance characteristics, and based on different current impedance characteristic, they classify different spores. So suppose if you have in the field a mixture of spores from different fungi, so then this system can detect, okay, so this spores is this much, this one, this much, and that sort of system is currently being developed. There is a couple of papers available online. All right, that's all what I wanted to cover today. Hope I have done something, and thank you. And before concluding, I must acknowledge some people, my organization, for allowing me to do this guest lecture for you, and funding agencies like GRDC, Hort Innovation, the University of Queensland, Coffee, and the Queensland government for helping during my PhD, and GRDC and Hort Innovation for the research that I'm currently doing. And thanks, Sita, for organizing this wonderful event. Thank you, everyone, for listening to me. Thank you.
Thank you so much, Zakeel, for your presentation, for your lectures.
Yeah.
Zakeel, could you please exit the share screen?
I did it, isn't it? Not yet? Wait 1 minute. I'll do that.
Okay, thank you.
Give me a second. Why is that not coming yet?
Not yet.
Not yet, yeah, not yet, I know. Yeah. My screen is sort of frozen. Give me a second. Yeah. Because I've got so many screens opened, and yeah, that may be causing. Yep, 1 minute.
Okay, no worries.
Sorry, yeah.
Okay, I
wait,
hope
wait.
while we are waiting you to close your screen, I hope the
Yeah.
student also prepared the question.
Yeah.
Okay, good. Thank you so much. Okay, Zakeel, actually we have one question from YouTube. Bentar, Zakeelnya ke ini ke close tadi. Sebentar lagi akan kembali. Oke, berikut di kolom chat ada link absensi. Silahkan Bapak Ibu yang hadir untuk mengisi daftar hadir. Kita tunggu sebentar lagi. Halo Pak Wid, ini ada Pak Widodo. Mungkin nanti kalau ada pertanyaan silahkan Pak Wid.
Sorry for that.
No worries, Zakeel.
Yeah.
Hang on. Wait a minute. Okay, we have a question from YouTube from Nurfianto. Question to Dr. Zakeel. How to measuring the disease severity, monitoring the pathogen, and predict the yield loss when applied to integrated disease management for area-wide management. Thank you.
Sorry. Can you repeat the question again?
Okay. How to measuring the disease severity, monitoring
How to measure severity? How to measure?
Measuring.
Measure. Okay. Yeah.
And predict the yield loss when applied to integrated disease management for area-wide management.
Well, yeah, it depends depending on whether, yeah, you can use simply you can develop a model to do this. But again, I don't understand what you mean by area-wide management. So if area-wide management is, so when you look at the, the control, so whether it is IDM or IPM or whatever, so the control and you look at the impact on a larger landscape scale. So you can still develop a model, which is what I'm currently doing here in a couple of projects. Where what you can do is we can estimate the disease severity or using AUDPC, and also in the model, if you can capture the green leaf area, which you can, use it as a proxy to your yield, so that you can determine your yield loss or whatever, So it's possible, yeah. So you can use sort of modeling approach. Suppose if you have all the parameters that you can use to parameterize your model, you can use a modeling approach. Or otherwise, if you are interested in carrying out an empirical trial, again, it is possible. But again, it's a lot of work. You have to establish a large scale field trials over, extended areas, many fields. And then you have to manually, do your disease assessment. The other option with area-wide management and then and with empirical studies, you can utilize the drone imaging options. So we can use drone images to classify disease severity and estimate disease severity, and then based on that we can use some machine learning models and then predict yield loss and so on. Yeah, there are various options available. So there are sort of machine learning models or approaches, and also other simulation approaches or other empirical studies. Yeah, hope I answered the question.
Okay, thank you so much. So for the second question, it will come from my student. Hang on, I will give the mic.
Yeah.
Hello, Mr. Zakeel.
Hi.
Thank you for the lecture. My name is Sidi. Right now, I'm taking master degree in pathology. And I have a few questions for Dr. Zakeel. The first question is it SIR model or SEIR model that you tell us earlier can be used on bacteria or virus or nematode as a plant pathogen? And then the second question is it, I'm interested about this portrait that you call BioScout, I think. So is BioScout can can identify directly the spore that trapped in that dose? Thank you, Dr. Zakeel.
Thank you for this question. The first question was, if you are working on a viral disease, the best model to go for is SIR or SCIR. Is that the question?
Yes.
Yeah. So it all depends, depending on your preference. But basically, SIR model is the very basic, simplest model, which we can use. But the SEIR is a bit more complex because you have one more compartment. But what people now prefer is SCIR model because the latent period is also an important component when it comes to disease epidemic, because for some, you know, pathogens or strains, the latent period may be shorter or longer, and also the latent period depends on the host species, right? And also the latent period is important when it comes to evolutionary epidemics. So for example, if you keep on spraying, so sometimes what happens, the fungicide, if you spray fungicides, when it comes to fungal pathogens particularly, so what they do is, so if they have an extended latent period, so what happens is if you spray, what they do is they sometimes shorten their latent period and then start sporulating very quickly. So that means from the latent period that move onto the next compartment that is infectious, so that they can produce their next generation before being killed by the fungicide. Did you get what I mean? Because that is the natural way of evolution. So that, so 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 if you spray fungicide regularly, sometimes evolution is, um, sort of, triggered, and then, what happens, that's one way. I mean, the latent period is shortened, and also the breeders' latent period is important because they always target to extend the latent period. So thereby you can, extend the disease cycle, so that you have, they have pathogen within the plant, but it doesn't do much to the crop, no disease express. So you pass a lot of time. So thereby, so for example, if you spray 5 fungicide per season, so if you have a very extended latent period, so then you can reduce, the infectious and period, and then the number of disease cycle if it is a polycyclic pathogen, and then number of sprays that are required to control the disease are reduced. So it's important. Latent period is very important in epidemiological, evolutionary, and also breeding aspects, perspective. So most of the modelers now, they always now go for a CIR model. And the second question is the spore trap. You said whether it is automated. Is that the question? So it's automatic. Detection is automatic. Is that the question? Yeah. Yes. It's. I mean, traditionally, conventional spore traps are just traps, and then we bring the spores here, and then we use microscopy to see it, and then we use molecular techniques to identify the strains or the pathogen, because there can be mixture of spores. There can be spores from different fungus. Some may be pathogenic, some may not be pathogenic for your crop. But now, so in those examples that I mentioned to you, so one is currently being developed where they are going to use a molecular diagnostic technique based on qPCR to have it there, because in the field, on farm, the spore trap collect the spore, and then using a chemical procedure release the DNA, and then the DNA is uh, inserted into another vial where qPCR will be done, and so on. So which is currently being developed. The second one is the BioScout one, which is already available, which is based on the morphology of the spores, microscopic structure of the spores, and a machine learning algorithm. So what they have done is they have developed an algorithm, machine learning algorithm, um, for various spore types of different fungal strains. So that these algorithm can precisely identify the spores that are coming in and give the signal, so how much of what strain. And that information is, yeah, given to the farmers. And the other one is in China, which is currently being developed, which is for different, fungal spores, how the change in the AC impedance happens. So based on these electric, you know, conductance and on resistant features, characteristics, they classify the spores, right? So then when the spore comes into these and these characteristic, electrical characteristics are recorded, and based on the previous, the, the, you know, the features, the spores are, identified and their concentration is reported. That is also still being researched. Okay.
We have a second question from my student. Hang on. Hello, Mr. Zakeel. My name is Adiba, and I would like to ask some questions. The first one is, what do you think to what extent an epidemiological model can be used to make a reliable prediction about future epidemics, and how can the accuracy and the reliability of
Reliability, yeah.
reliability of epidemiological models can be improved more, in your opinion?
Yeah, well, yeah, I mean, yeah, there can be different types of epidemiological model. For example, if it is a weather-driven model, so there are some weather-driven epidemiological model in which we can just predict the risk. Say, for example, I can't remember the name of that. So for different, so there is one company in the US, they have developed some models for various pathosystems. So we can develop an epidemiological model for the pathosystems. So based on whatever the models that I have already discussed. But again, these models, so, the epidemic, as I said, it depends on other factors like, host and environmental factors. So then what happened is, even though there is a pathogen and susceptible host, and there should be a conducive environment. So then we have to put weather parameters there in the model to predict future epidemics. So that's a prediction model. So we call weather-driven prediction models. There are a lot of weather-driven prediction models available already, so which you can develop a weather-driven prediction model for your pathosystem using an available a disease model or a new model, and then the weather parameters there. So the accuracy and precision all depends depending on how well the model fits your data that you have, right? So then, yeah, I mean, yeah, not all the model are perfect 100%. So there is a bit of, yeah, the concern about the precision and accuracy, but at least we can increase the precision of the model by fitting the model well. And if you have plenty of data, of course, we can fit the model very well. Otherwise, these models are sort of quite useful to scenarios where you try to compare different interventions or different management options. For example, one student or somebody asked from the UQ that how we can use this model to sort of, predict the outcome of area-wide management. So of course, we can use these epidemiological models to see, which intervention or which management option is doing better. Yes, yeah. Hope I answered your question. Yeah. But remember, not all the models are good. Yeah, I mean, yeah, not all the models are good models, and it all depends depending on how you refine it and how do you, yeah, and also your parameters. So when you are developing a model, a simulation model, for example, so how well you parameterize the model. Okay, so it all depends. Then define the parameters, whether you really do establish an empirical study and develop the parameters, or you just find the parameters from literature and how well those literature report those parameters, and all depends, right? So all these contribute to your final precision and accuracy of the prediction. Yeah, I didn't hear.
Yes, Zakeel, thank you so much. Is there any
You're welcome.
question? Okay, one more. Is it okay? Is it not too late for
Yeah.
you?
Maybe the last question. Yeah.
Okay, good.
Yeah. Yeah.
Thank you so much, Mr. Zakeel. I am Fatima. I want to ask you, how do different epidemiological modeling approach, like mentioned before, SIR, SEIR, account for factors like population heterogeneity or seasonal indices transmission? Maybe that's my question.
I didn't hear the last part of the question. How the SCR, so SIR or SCR models?
Yeah.
What's the
Uh, account for factors like population heterogeneity or seasonality in disease transmission.
seasonality and
In disease transmission.
disease transmission?
Yes.
So what's the question? How the model?
How do different epidemiological modeling,
Yeah.
like SIR,
Yeah.
account for factors like population heterogeneity or
Yep.
seasonality in disease.
Yeah.
In
Well,
disease.
yeah. Yeah. The population heterogeneity, I hope you are referring to the host heterogeneity. Yes, of course, we can account for, because I have developed one, we not I, we have developed one epidemiological model we call LANDSEPI with our colleagues in France, where we can account for landscape heterogeneity and also the host heterogeneity. For example, if you have, say, what proportion of the landscape is occupied by resistant host, and what level of resistance, and what proportion of the landscape is covered by non-host, what proportion of the landscape is covered by susceptible host. Yes, we can account for that in the model, and accordingly we can find the epidemic outcome. And also in the model that I said, we can find some evolutionary and economical outcomes, as long as we have parameters for economic and evolutionary parameters. And yeah, it's possible in the model to account for the heterogeneity, I mean the landscape and the host heterogeneity. And also the other one is the seasonal. The seasonal, again, it depends depending on the type of model that you're using. If it is a weather-driven model, of course, yes, the factors, you put your parameters are from your weather parameters. Or some epidemiological model like, the one that I said, Landscape, where, you have to, it is not a weather-driven model, but the parameters that you calculate to parameterize the model are based on the weather factors, right? Suppose if you are developing a model for the particular pathosystem under particular environmental conditions, yes, that is accounted for. But if your parameter is not, then it is not. Okay, it is very contextual. It's based on how you determine your parameters for your model. Yeah. But if it is a weather-driven model, yes, of course, it is one of your input for your model. Yes. So it all depends depending on the type of model that you're going to use or the type of model that you're going to develop. Yep.
Okay, thank you so much, Zakeel.
You're
Okay,
welcome.
probably because it's too late in Canberra, so before I close this guest lecture, if students want to ask some question to Dr. Zakeel, you can meet him in person in November.
November.
So he
Yeah.
will come as a keynote speaker in the third conference of CSIRO. So I hope the student also can join in the conference. And
Yep.
if you want to talk or ask further question, you can meet him in
Sure.
Bogor. Okay. Okay, Zakeel, before we close the guest lecture, we would like to give you the certificate of appreciation. This
Thank you. Yeah.
certificate is presented to Mohamed Zakeel Mohamed Cassim, PhD, as speaker in our guest lecture, Plant Disease Epidemiology, Case Studies on Tracking and Managing Pathogens Spread Across Landscape. Uh, we will give you online, so please receive Zakeel, and we would like to send the certificate through email.
Thank you.
And thank you so much. And for the students, see you on the another guest lecture. And good afternoon, Zakeel.
Good
Have a
afternoon.
have a good weekend, everyone.
You
See
too.
you. Bye.
Thank you. Bye.
Thank
Bye.
you. Bye.